Our next speaker will be Alan Jasanoff from MIT, from the Department of Biological Engineering. And he will speak to us about molecular probes for non-invasive neuroimaging.
Well, thank you very much, Ralph, and thank you to all the organizers for what I think we can see is already a great symposium. I'll try to live up to that. So the problem that I work on is the problem that probably almost everyone here works on, which is trying to somehow tackle the complexity of the brain, understand brain functions in particular at a systems level, or at least that's my concern. And we're limited by technology, and that's why we're here today, here now. Most neuroscientists face a choice between very specific measurement techniques, electrophysiology being, of course, the most famous, typically invasive in animals, optical techniques now providing more coverage than electrophysiology techniques, both providing unparalleled specificity, but relatively little coverage of all these billions and billions of neurons. On the other end of the neural recording spectrum, we have techniques like bold fMRI, blood oxygenation level-dependent functional magnetic resonance imaging, which is the standard brain imaging technique that people use in humans, and that's generated thousands and thousands of papers and enormous insights into the function and organization of the human brain. But that really isn't the best technique for studying neural mechanisms, and that's because when we see fMRI data, typically in the form of sort of blobs of what's generally referred to as activity, we often don't really know what's going on underneath those blobs. What cells are active, how are they interacting with each other, what chemical and physical players are involved. So these techniques really provide whole brain coverage, unparalleled sort of comprehensiveness, but very limited specificity and resolution. And so my lab set out, perhaps foolishly, to try to find some sort of middle ground between the specificity of electrophysiology and optical imaging and this whole brain coverage, and we turned to something called molecular imaging. Specifically using MRI contrast agents that sense neuronal activity. So what is molecular imaging? Well, it's the combination of molecular probes that detect molecular level activity with generally non-invasive imaging techniques. And we can look at a few different areas in which molecular imaging research takes place. One of the most famous is something called positron emission tomography, or PET scanning. MRI is a newer approach for molecular imaging, and of course optical imaging is used in various different ways to perform molecular imaging. And these techniques differ in important ways. So sensitivity is one of the ways that they differ. PET scanning has femtomolar sensitivity, and actually for many years PET was really the only way to do molecular imaging. There was a Society for Molecular Imaging that is basically a PET society because of this enormous sensitivity. In contrast, MRI has micromolar sensitivity, which is considered pretty pathetic by many chemistry standards. Optical imaging, a bit better with submicromolar sensitivity, nanomolar sensitivity in many cases. So MRI doesn't stack up too well that way, but it has an important advantage compared with PET. Both MRI and optical imaging have much more versatile capacity for sensing and faster dynamics. And that's largely because PET is performed with radionuclides. It's based on radioactive emission of the PET tracers. They can't be turned on and off. And the physics of PET probe detection limits the resolution of those techniques to about sort of millimeter scale. Often in practice, it's actually more like several millimeters or centimeters. The temporal resolution is set by the biokinetics of PET probes, and so that's also hugely limiting, typically to the order of minutes. So we can do a lot better with MRI or optical imaging, and we can do so well with optical imaging that one might be tempted to work with that. But the problem is that we can't get the whole brain, at least not yet, not in the kinds of animals that we find most cuddly, larger rodents, humans, monkeys. We won't be able to access the whole brain. MRI, in contrast, is non-invasive, and it provides whole brain coverage. And so there is actually a strong reason to try to develop this technology around MRI contrast agents that sense neuronal activity to try to break past the non-specificity of conventional brain imaging techniques. So that's what I'm going to talk about today. First, let me tell you just a little bit of introduction about MRI contrast agents. What are they? They're animals that might not be familiar to many of you. They work fundamentally differently from molecular probes for any of these other techniques. They're mostly based on magnetically active molecules. This is a quintessential example here. It's gadolinium DTPA. It was the first clinically approved MRI contrast agent. And it's built around this atom of gadolinium, a lanthanide that's paramagnetic and that couples magnetically to the nuclear magnetic spins in water molecules. Water molecules are the basis of most MRI. When we get MRI images, we're imaging water, and as Jack D. Ripper, for those of you who like Dr. Strangelove, observed 70% of us are water, so it works pretty well. On the other hand, these contrast agents don't add a new color. They change the magnetic behavior of water molecules, and they either brighten or darken MRI signal. There are so-called T1 MRI contrast agents and T2 agents. They work by slightly different mechanisms that I won't go into, but roughly speaking, the T1 agents brighten MRI signal, the T2 agents darken the signal in proportion to the amount of the contrast agent added, and that's also influenced by the way that the imaging is done. And the beautiful thing about this, and of course this is an image in a microtiter plate, the beautiful thing about these things is that they work in vivo. So these are images of a rat that had a T1 agent injected into its eye, and you can see that the eye is lit up over here. The eye is actually okay. It got a small dose of manganese, which is an example of a T1 agent, and the manganese was actually transported from the eye to different brain regions, something called the thalamus and something called the superior colliculus, that both light up because they got a little bit of dose of this contrast agent. Now, my lab is interested in not just illuminating anatomy in brains, but actually in modulating the contrast due to these agents to reflect the activity of the brain itself. And so I'm going to tell you in the rest of my talk first about a maturing project on molecular imaging of neurotransmitters in vivo that uses a contrast agent that we began developing a number of years ago. And then I'm going to tell you about a much more exploratory project to try to boost the sensitivity of molecular detection using MRI to get into the sort of sweet spot that optical imaging can detect. So let me start out by talking about molecular imaging of neurotransmitters, and I'll tell you about functional imaging with dopamine release and also neurotransmitter transport detection. Many of you know about, neurotransmitters, of course, are chemically distinct molecules that allow neurons to communicate with each other, and they have functionally distinct roles. And one of the neurotransmitters that's gotten a lot of interest over the past couple of decades is dopamine. It is the reward neurotransmitter. It's closely associated with addiction and as well as the control of movement. Pulses of dopamine release in parts of our brains as well as rats' brains seem to reflect aspects of learning and motivation. The dopamine is most famously a neurotransmitter that allows animals to learn reinforcement tasks like Skinner box tasks, lever pressing for reward. So we were interested in whether we could actually develop a three-dimensional mapping technique to study how dopamine functions in reward-related paradigms. And so we decided to develop a sensor based on a protein. We collaborated with Francis Arnold at Caltech to engineer a protein called P450BM3 to act as a specific sensor for dopamine. This protein is a an MRI contrast agent because it has in its heart a heme, a paramagnetic group similar to the heme in hemoglobin that allows the contrast agent to change MRI contrast. And it also binds small ligands right near the heart of right near the heme. And so we wondered whether this could actually be used as a ligand sensor. Unfortunately, it doesn't naturally bind dopamine. It naturally binds something called arachidonic acid. So when we started the project, we first took the protein, we measured a parameter called relaxivity, which is like the quantum yield of an MRI contrast agent, and we challenged it with AA, arachidonic acid, and we saw that in fact the relaxivity changes, the contrast in an image changes, and actually if we dump huge amounts of dopamine on the protein, we could get changes as well. And so in vitro, we could see these relaxivity changes, and they translate into image changes. Each one of these ligands turns off the contrast agent, or tends to turn off the contrast agent. That's why you see a darkening. Now, we wanted to get better sensitivity and specificity for dopamine, and so we, working with the Arnold lab, performed directed evolution to try to engineer better specificity for dopamine. That succeeded over several rounds. We were able to get micromolar sensitivity for dopamine, this is log KD, over rounds of evolution, and we also lost sensitivity to the natural ligand arachidonic acid. And of course, we did a lot more work to show that we have adequate selectivity for the neurotransmitter. So at the time, it was a new approach to generating MRI sensors, and these were the first MRI sensors for dopamine or any neurotransmitter. So how do they work? In the heart of the protein, as I mentioned, there's a heme. When dopamine binds, it blocks access to the heme. It coordinates directly to the iron atom at the center of the protein, displacing water molecules that I didn't show. And that interaction turns off the contrast agent. It prevents the interaction that physically underlies the contrast effect that this protein creates. This is work that came from crystallographic studies of Erik Brustad, published a couple of years ago. We did this work. A lot of people were actually still quite skeptical that we could actually use this thing for anything useful in vivo. And sort of foolhardy as we were, we actually went on and we tried to perform real functional imaging in 3 dimensions using these probes. And we did this using a sort of relatively crude paradigm where we inject the sensor directly into the brain in an area where we knew the dopamine signaling was important, an area called the ventral striatum, which is the primary target of dopaminergic neurons that originate from the midbrain in the structure called the VTA. We injected the contrast agent. We stimulated a fiber bundle called the MFB in the lateral hypothalamus, which is a very famous, well-known rewarding stimulus that was discovered by Olds and Milner in the 1950s and is the basis for addiction paradigms that have been very useful in quantifying the efficacy of addictive drugs. So we asked, could we map dopamine release under this stimulus? And our paradigm we delivered some drugs, L-DOPA, to prevent dopamine from being depleted in our experiments, and GBR12909, which is a dopamine reuptake inhibitor. It skewed the results in our benefit by allowing dopamine transients to last a little bit longer. These were first of their kind experiments, and so we wanted to keep the odds in our favor. This is the way our experiments ran. So we pre-inject the sensor for about 45 minutes. That's essentially to load up the brain with this thing. And then we start giving stimuli. We gave repeated stimuli in what's known as a block paradigm. It's kind of similar to how a lot of fMRI experiments work, conventional functional brain imaging experiments work. And what we were expecting was that as we inject the sensor, we see an increase in signal. That's because this is a T1 contrast agent, boosts the signal. But then as we give the stimuli, if there really is dopamine release, which there must be, we should see little dips in the signal, little transients that result from the neurotransmitter turning off the sensor and then, of course, going away and allowing the sensor to recover. We put this into practice. And here is an image of a rat that has a cannula implanted in its ventral striatum. It's slightly unphotogenic, but it works. And you can see that around the tip of the cannula, there is brightening. And the brightening is because of the contrast agent that we infuse. So it, again, doesn't add a color. It increases the signal. And we looked at the signal in this region of interest over multiple cycles of stimulation. So could we see those little dips? And what this shows over here is a temporal trace over multiple stimuli in a single animal, averaged over a region of interest like this. And in fact, you can see that there are these little dips of about a couple of 1 to 2% of MRI signal change as we stimulate the animal. There are a lot of things that go on in brains. One of the things, hopefully, is what our contrast agent is doing, but we worried about bleed through of hemodynamic effects and various other things that could be going wrong. And so we have actually quite a nice control for these experiments here. We compared the ability of the MRI sensor for dopamine to detect signal changes with the ability of a control protein that was exactly the same but just didn't have the mutations that conferred dopamine selectivity. So we don't have a catchy name for the dopamine sensor, 97, volunteer names for me in the question period. You can see over here the impulse response function for stimulus sensitivity in the presence of the sensor. So this is essentially averaging over multiple stimuli, and you can see there's, again, this pretty robust signal change, whereas with the wild-type control, it's more or less a flat line, no significant signal change. And that shows that, in fact, the dopamine binding property of this protein is required for the observed signal changes. Now, how well do the signal changes that we measure compare with a known established measure of dopamine release? We did electrochemistry using amperometric electrodes in the same region of the brain here. And in fact, we could detect little dopamine transients due to each stimulus. The signal to noise is much better. Of course, this is a point measurement. And they followed more or less the same pattern that we could see by MRI, though less noisy, of course. We could use the electrochemistry actually to prove that the dopamine sensor in the brain of the rat is binding dopamine and behaving according to the mechanism that we expected. So this is a trace, an amperometric trace that we obtained in the absence of the sensor, and this is in the presence of the sensor. So these data are part good, part bad, like probably most things in the world. And they're bad, of course, because of, as you can see, the presence of the sensor buffers the dopamine. It damps down the dopaminergic transient that we can see over here. On the other hand, it proves that the sensor is binding dopamine. Now, one thing that I can note is that these amperometric traces actually are not reflecting specifically synaptic dopamine. They're reflecting probably primarily dopamine overflow from synapses. The dopamine concentration in synapse is probably more like a millimolar compared with the micromolar levels that we're detecting here. And so we think that the sensor probably isn't having a huge effect on synaptic dopamine, despite its perturbation of the system. All right, so the addition of the sensor buffers dopamine. It proves that the sensor is binding in vivo. The wild-type protein doesn't bind dopamine in vivo, and in fact, that's also backed up by the amperometric trace here. So it proves the mechanism of the contrast agent and shows expected signal changes in vivo. All right, now could we use this for actual brain mapping? That was just a region of interest analysis. It shows that detection is possible. These are the results of averaging signal changes over a collection of animals, 7 animals, that all got the dopamine sensor injected into the ventral striatum and got these stimuli. Now, there are a few problems with this. So one is that every injection is different. So for those of you who do brain injections, you know that, we like to imagine there's a sphere of the agent propagating from the tip of the cannula, but that's actually rarely true. Every injection is different. Moreover, the injection, although it covers a pretty big chunk of brain, several cubic millimeters, it does not cover the whole brain. The contrast agent falls off, and as it falls off, you lose the ability to distinguish between no dopamine and no contrast agent. So how could we solve those problems? We noticed by looking at the properties of signal change in a model, in a mathematical model of how these things should work, that as long as we don't saturate the sensor, there is a roughly linear relationship between the calculated percent change in MRI scans and the dopamine concentration that is released. So, of course, each one, at any place where we have our dopamine sensor, we are going to saturate it at some point. If we have 20 micromolar sensor injected into the brain, then if we have well over 20 micromolar of dopamine released, well, we're not going to be able to detect that. And in cases like this, we'll get nonlinearity in our signal. However, if we can determine a priori where we have sufficient sensor to perform measurements, we can use the slope of this line to convert MRI signal changes back to quantitative concentrations of dopamine detected by the sensor. And so what this inspired us to do was to segment each one of the brain data sets that we got from our individual animals based on the amount of sensor that got into different parts of the brain. We could do that because we monitored the MRI scans as the sensor was being injected, so we could measure, well, what is the signal change due to the sensor injection? And so we do the segmentation, and then we average over voxels that are emitted from each one of the animals. So using that approach, we could get a quantitative measure of dopamine, and that's shown over here. So these are now close-ups of 3 coronal scans, so this way zoomed in on the ventral striatum, which is, again, this area where we're targeting our injection. It's also the area that gets peak dopamine release. You're seeing the atlas, brain atlas overlay in white, and in the blue outline, which probably many of you can see, shows the voxels that pass the injection threshold criterion. And you can see that there is actually a pretty decent-looking spatial map of signal change here. It's on a quantitative scale in terms of concentration of dopamine, and that's because we were able to use this analytical method that I pointed out. What is the signal to noise here? Well, of course, there is variability over animals and over experiments, but nevertheless, we can quantify the signal to noise by calculating mean and standard error across sort of cross sections of this. So imagine, an image with error bars around it in 3 dimensions, but we're taking cross sections, and you can see that many of the features that these lines pass through are actually sort of, reasonably robust when you look at the signal to noise in this way. So we can get 3-dimensional quantitative mapping of dopamine, and we can see features. So I think you can see that there is what appears to be a focus of dopamine release in this area called the nucleus accumbens core, which is a key area. It had been identified as a focus for dopamine release in experiments by Mark Whiteman's lab, who's here, and others. And of course, this pattern is dependent on the conditions that we use for our experiments. Nevertheless, I think it shows that you can get spatial architectonic mapping of neurotransmitter release. This focus of dopamine release may be specifically significant for the paradigm that we used, but we need to do more work to figure out, how specific it is to the conditions that we used. The signals are quantitatively similar to electrochemistry. So these are, quite different measurement techniques. One using an electrode that's buried in part of the brain, measures dopamine overflow. In this case, the MRI probe goes everywhere. It's detecting synaptic dopamine in principle, dopamine overflow, and distal dopamine from the synapses. And yet the measurements are actually quite similar in terms of actual sort of micromolar. We can look over 3-dimensional volumes. Obviously, that is the point. And we can look fairly far away from the area where we injected the sensor. So dopamine release in ventral striatum is actually relatively well studied, but we could actually see hints of dopamine release in an area called the reticular nucleus of the thalamus. It's a sort of lateral zone of the thalamus that is known for expression of D4, dopamine 4 receptors. And we could see signal changes there, again, relatively quantitatively with error bars and so on. So dopamine transients can be observed outside traditionally studied hotspots. We can see dynamics. Now, this is nothing like the kind of movies that you saw in the previous talk. It's vastly sped up, so each frame is about 8 seconds, but you can see the rise and fall of dopamine over individual frames in the movie sequence. This is averaged over multiple stimulus cycles, so it's essentially the impulse response in 2 dimensions here, of dopamine release due to the stimulus, we can actually look at quantitative features of how these dynamics work. So we divided up the ventral striatum into different regions of interest, nucleus accumbens, core, shell, and various other regions. And for each one of these, we can get a time course. We can fit it to a model of dopamine dynamics. So this is a single compartment model that reflects dopamine release and dopamine reuptake. And from that model, we get parameters for dopamine removal rate and release rate. And we can see that at least under these conditions, most of the diversity among these different regions is reflected in release rate differences. So the pattern that we saw with focused dopamine release in nucleus accumbens core is because of actual increased release there as opposed to differences in reuptake. Again, we're using drugs, slightly artificial paradigm, but this analysis at least can be done and shows that the single compartment model fits the data with the highest release in the nucleus accumbens core. Can we actually learn something about the neural systems that give rise to these patterns? That's really where we want to go with these measurements. And we took a very sort of amateurish stab at this by comparing the electrode sites for our stimulation experiments. Well, we targeted a particular set of brain coordinates, but nothing's exact. You always get a little bit of range. And we compared that with the centroids, the sort of the centers of the molecular imaging result. And we could see, so each individual animal has its own color, and there's basically no clear mapping there. So, the most obvious explanation for, well, why do you get a focus over here is, well, because your electrode is over here, and its fibers project to the place where the hotspot was. That didn't seem to be supported by these data, but of course they're noisy, so there may be more work to be done here. We also looked at the stimulation coordinates versus the amount of dopamine release that we observed. And we could see actually what turned out to be a pretty strong dependence between the rostrocaudal coordinates, in other words, front to back, of the electrode position and the amount of dopamine release that we saw. So, and this is really over a fairly narrow range. So it's about 1 millimeter difference makes actually a few-fold difference in dopamine release. So it suggests that local fibers near the area that we're stimulating actually contribute to this result. So it's a stab, as I say, at part of the mechanism that may give rise to the peak dopamine pattern that we saw. Response amplitude but not position seems to depend on the precise stimulation site, but really what we want to go on with this to do, and this is ongoing work in the lab, is to combine this molecular imaging with a sort of a systems-wide approach that's based on this less specific but more comprehensive regular functional imaging, the bold fMRI, which is, again, the basis of human functional imaging. Can we actually relate the pattern of dopamine release to whole brain-wide patterns of activity? All right, so I've told you in detail about work that came out in a paper last month. It turns out you can use these sensors for other things. One obvious thing you can do is, make sensors for other neurotransmitters. So we made sensors for serotonin as well. And we're using these to study serotonin transport in vivo. So serotonin, stimulus-evoked serotonin impulses are a little bit smaller than dopamine impulses, and we haven't yet been brave enough to try to map them in 3 dimensions. But many of you know that serotonin transport, and in particular the process of serotonin reuptake from tissue and from synapses, is a critical aspect of affective control in humans and animals. And so we did experiments where we tried to, where we injected a serotonin sensor with the neurotransmitter. So we're bringing our own serotonin to the game, and then modeled signal changes over time to try to measure serotonin reuptake using the sensor. And so we did this in many animals, and we applied to the animals a compartmental model. It's a little bit like PET, but here we're actually looking at the differences between we're looking at the differences between intracellular versus extracellular serotonin, probably first and foremost, which is something that actually you couldn't see using a radionuclide-based technique. So we set up this compartmental model, and we used it to model the dynamics of sensor injection in the presence of serotonin. So we're injecting into striatum again. This is an example of an animal with a field of view denoted here, and I'm going to show you a little movie of how the model here corresponds to data over an injection. you can see, I think, that the model actually does a pretty decent job replicating the spatial pattern of signal change that we see in actual data. And that shows that that suggests at least that the parameters of transport could be actually pulled back out of this model and used for quantitative mapping. We have preliminary versions of that over here. The black voxels were not included in the analysis because they had corrupted signal of various sorts. But what this shows is a map of unbinding rate, or the sort of, in a sense, the rate constant that converts the sensor bound to neurotransmitter to the free sensor that gives rise to the signal change. You can see there's, again, some evidence of structure, and we've got to go further with this. This is work of Aviad Hai. Okay, so compartmental modeling yields rate constant estimates. It's a different way to use the sensor, the type of sensor that we created. All right, now there are obviously many aspects of the experiments that I showed you that are crude, and one of the most obvious ones is that, well, we had to inject the thing into the brain. So, non-invasive imaging is only as non-invasive as your cannula. So can we actually deliver the sensor in a less invasive way? Well, one of the chief techniques that we're trying to use is uses ultrasound to perform non-invasive trans-blood-brain barrier delivery. So many of you know that the blood-brain barrier is the chief impediment to especially large molecules that you try to get into the brain systemically, and we're interested in delivering them through the bloodstream. If you sonicate part of the brain, you can transiently open the blood-brain barrier and then deliver contrast agents peripherally and get them into the brain. And so this shows an example where we delivered gadolinium DTPA. This is a coronal section through a brain. You can see part of the brain is accessed here with the contrast agent, and that's without, of course, direct injection. So no surgery was involved. We obviously want to get bigger fields of view. That means bigger transducers and so on. We have totally different projects in the lab that try to get around the delivery problem in different ways. One of the chief directions is to develop cell-permeable contrast agents, and this is work that we have as a long-standing collaboration with Steve Lippard's lab at MIT. And then lastly, we're quite interested in genetically encoded MRI probes, where basically, at least for animals, these things would be expressed inside brains, and you could detect signal changes with them, and we have a couple of papers on this already. All right, so now, these are all incipient technologies. They are going to work to varying extents at detecting, at mapping neuronal activity at molecular level, in vivo in many cases, we hope. The chief limitation is actually not the delivery of these agents. The chief limitation to, let's say, discovery-oriented neuroscience is actually the sensitivity of these things to both detection by the imaging technique and also to the molecules that we're trying to detect. So we're very interested in ways to qualitatively improve the sensitivity of MRI detection schemes. And I'm going to tell you about one of these ways right now, and this is an idea of manipulating the biological system itself to try to non-invasively reflect its own activity, and something that I sort of tongue-in-cheek refer to as molecular imaging using synthetic biology. So one of the most obvious ways that one could try to boost sensitivity for an insensitive technique is to use an enzyme. And this is a tried and true approach in optical imaging and other contexts. We've dabbled in this as well. So we did work where we expressed an enzyme that processes an MRI contrast agent. So the biological signal itself is the enzyme, and it doesn't have to be there in huge quantities because it processes the contrast agent. And we showed that if the enzyme is extracellular, well, it alleviates the delivery problem somewhat. And you can use this thing to basically map in at least preliminary experiments, patterns of gene expression, patterns of the enzyme expression in MRI scans. This is an ex vivo image where we stained for this particular enzyme, SEAP. We also did this work in vivo. We could show that virally driven enzyme expression can be detected by contrast agent that's been infused. And I won't actually go through this in detail, but it gets around the problem of low amounts of a signal by using a signal that amplifies itself, the enzyme. But it has other problems, right? So we still need the contrast agent. This is fundamentally the source of the signal that we're detecting here, and it still needs to be present in micromolar quantities. It still would need to get into the brain somehow. In this case, we did invasive delivery, but we could use ultrasound and so on. But it's still a limited technique. We need to deliver an exogenous substrate, and there are problems with kinetics, signal to noise, still possible, of course, because of the complexity of the mechanism. So is there some way to simplify this? Well, an idea that we had was actually inspired by this bold fMRI technique, the very technique that, in a sense, we set out to circumvent or improve on. And that's the idea that the blood vessels are themselves actually a great contrast agent. They have 8 millimolar hemoglobin in them. Hemoglobin is a paramagnetic molecule. It's because of that molecule that you can do functional imaging at all using MRI, at least in most of the ways that this technique is used. Now, these are a set of neurons in part of the rat's cortex. These are a set of blood vessels in a comparable region, and you can see that the blood vessels kind of embrace, or they snake around the neurons. They are in a rather close embrace. It's obviously not, one neuron, one capillary, although people who study this for a living might argue that in some cases there is almost such a close correspondence. So is there some way that we can transduce the signal from these neurons into signals in the blood vessels without losing all specificity? So in normal functional imaging, there are a set of biochemical pathways that bring neuronal activity to the vasculature and that enable neuronal activity to be kind of nonspecifically detected. We can't tell, for instance, the difference between excitatory activity, inhibitory activity, the results of different neurotransmitters, and so on. Can we make that more specific? And so this is my little icon of this. This is a neuron under normal circumstances sending a nonspecific signal to the vasculature. Could we actually invent or introduce artificial molecular players that make that interaction more specific? If we could potentially achieve a much greater level of specificity than regular functional imaging, as well as much greater sensitivity than we can now access using our conventional MRI contrast agent. So this is really a pretty new concept for molecular imaging, and it's something that the geeky name is engineered hyperemia, so using specific molecular probes to control blood flow. And one of the vaguely cool things about this, I think, is that although of course it's slightly messy because it makes use of physiology mechanisms that are inherently complex, it can translate across a broad range of imaging techniques that have different strengths and weaknesses in space and time. So we can potentially detect the signals using optical or ultrasound methods that are more portable, maybe suitable for awake behaving animals, and faster. So I'm going to tell you about one example where we used the most potent vasoactive peptide, or one of the most potent vasoactive peptides, something called CGRP, that acts at nanomolar concentrations to achieve signal changes at a molecular level, but using this vasoactive mechanism. We injected 500 nanomolar CGRP into an exposed rat brain prep and looked at vasoactive responses, and you can see that all over these different regions of interest. So this is a blood vessel here. This is parenchyma here. For each one of these regions, we could see signal dips that corresponded to that were produced by the injection of CGRP. Now these concentrations, 500 nanomolar, this is 1,000-fold less than what we use for our regular contrast agents in MRI. This is the concentration, by the way, in the tube. So once it gets out into the brain, it's diluted by a considerable factor, probably 10 to 100. We could see these changes by MRI as well. So here is a rat brain. It's been injected with CGRP on one side and artificial cerebrospinal fluid, the control, on the other side. And we can see a signal change of a few percent, 5%, focused in the region of the injection, nothing for the control. And very importantly, this signal change is dissociable from the normal mechanism for bold or endogenous hemodynamic changes that occur in functional imaging. So here we did the same experiment both before blue and after green, infusion of L-name, which is a nitric oxide synthase inhibitor. That inhibits or blocks the main pathway for bold signal generation in the brain. And you can see that there's very little signal attenuation in our engineered hemodynamic response here. So we do have something that's potentially very specific. And again, the sensitivity is 1,000-fold greater than conventional contrast agents for MRI. So for us, that's kind of significant. 1,000-fold, it's better than 2. What can we do with this? Obviously, that was a fairly dumb experiment where we're just taking the agent, injecting it. We can see it. Okay, well, that's what actually most medical imaging does with its contrast agents, but we'd like to go further. We want to actually detect things turning on and off. So this is quite new data where our concept is basically to regulate how these probes interact with the biological system. And the basic concept is to use blocking domains. So in green here is a, artificially conceived blocking domain that either blocks or doesn't block the activity of the blue thing, which is the CGRP-based probe. The idea is when it's not blocking, then we get our vasoactive effect. When it is blocking, we don't. And we can imagine at least different variants of this, where we turn on and off the blocking using something as crude as a protease, which just would cut them apart. That's pretty much bound to work, right? And potentially a ligand sensor. We could imagine having this be conformationally regulated in some way. We set up an in vitro culturing system to try to test these things. So we have reporter cells that are engineered to signal via the CGRP pathway by overexpressing the CGRP receptor. And so we can dump trial versions of these constructs on the system and actually try to engineer them. So it's sort of an indirect way to do the protein engineering that it takes to get the probe here. And we've started out by working with protease-based probes. It's the sort of low-hanging fruit, so to speak. We developed caged CGRPs that basically only activate the receptor at verging on millimolar concentrations here. On the other hand, when they're cleaved, when they're liberated as wild-type CGRP, they activate at around a nanomolar or even somewhat less, in some cases, concentration. Here is a set of constructs that we developed to sense different proteases. So in each one of these cases, we have the blocking domain, then a linker with the protease site, and then the probe itself, CGRP. And in each one of these cases, we're detecting the output from one of these assays, and we have plus-minus protease inhibitor. And you can see that one can set up systems like this that work with a few different examples of these protease inhibitors. So in other words, we can generate protease sensors for in vivo testing, and more importantly, or more generally, it's clear that there are ways to turn these things on and off, to regulate their function, and we can imagine going on and doing much more elegant and also neurally focused experiments with them. So this is work in progress. Let me summarize the talk. So I first told you about the first functional brain mapping using molecular MRI. Our probe revealed pattern dopamine release with a quantitative readout of neurotransmitter concentration. I showed you an example of MRI sensors detecting additional neurotransmitters, serotonin, and we use this to map, or we are using this to map serotonin transport parameters in vivo using the kinetic modeling approach that I told you about. And then I ended by telling you about the new concept of molecular MRI with engineered hyperemia. Now, obviously, there's a ton more work to do with this. This is, in general, this molecular imaging approach to studying neuronal activity is a pretty new development, and we have a lot of directions to improve on. We want to combine sensors and conventional fMRI techniques to actually push, let's say, the scientific contribution of these techniques to try to understand or situate the molecular level readout in the broader activity patterns. And this will enable us to do what's called functional connectivity analysis, we hope. That we have many improvements to make in delivery, sensitivity, and specificity, and I told you about one or two examples where we're trying to achieve this. And then, we need to bring this molecular fMRI technique with vasoactive probes a lot further along, and there are actually many, many directions to bring it, including genetic encoding as well as detection of neural activity parameters in various different ways. So with that, let me thank the people who did the work, and I'd like in particular to acknowledge Mikhail Shapiro and Gil Vestmeyer, who led the initial work with the dopamine sensor. That was done in collaboration with Francis Arnold, Eric Brustad, further refined the sensors and generated the serotonin sensor that was then used by Aviad Hai for the kinetic modeling. Taekwon Lee, in collaboration with others, did the functional imaging of dopamine that I spent most of my talk on. And then Adrian Sluserchik and Mitul Desai have led the hemodynamic fMRI project, the hemodynamic molecular imaging project. I'd like to thank funding and you for your attention.
This is a great direction to go in. What are the prospects in the future for more rapid kinetic analysis? The acquisition, I guess, at this point is on the order of 5-8 seconds, something like that. How do you see that improving?
That's right. So currently the dopamine sensor kinetics, we can't access super fast kinetics because we're acquiring image frames in every 8 seconds. On the other hand, that was sort of a considered choice. We want to boost our signal to noise. That's improved with longer image acquisition, but we also face a kinetic limitation due to the probes themselves. They have on and off rates that we measured using stop flow techniques, and they're on the seconds time scale. So the kinetics in our current experiments are distorted by that. How can we improve on that? Well, again, protein engineering, of course, presents itself as a possibility. We didn't select specifically for kinetic properties. I think we were right not to, actually. In fact, with that specific set of sensors, it may be hard to do it. But there are other probes that we work on that, in particular, calcium-dependent probes that don't actually require, as tight binding coefficients that will probably have intrinsically much faster kinetics. Now, how are we going to let the imaging catch up to that? There are a couple of different possibilities. So one is money. That's because with more money, you can get a bigger scanner, a higher field scanner, which has better signal to noise, allows you to acquire the same data in less time and get a faster frame rate. The other way is to make sort of trade-offs of the sort that, for instance, optical imagers are quite familiar with: line scanning, restricted fields of view. Imagine getting a low-resolution whole brain scan and then zooming in on a region and getting data there faster. There are also phase-locking techniques that we could use.
So MR spectroscopy has been used to probe molecular level interactions, biochemical interactions, at least at the level of muscle. And I was wondering if that's an alternative or another option for improving.
Yeah, so in a sense, MRI spectroscopy, which is really the original type of NMR or MRI, is the predecessor to any form of molecular imaging that can be done with these contrast agents. There are key differences. So typically in MRI spectroscopy, or MRS as it's actually usually called, we're actually detecting the spins that are attached, the protons that are attached to those specific molecules that we're detecting. There are usually several orders of magnitude fewer of those spins than of water protons, and that's why those techniques tend to be quite insensitive. If you look at a typical MRS experiment, the voxel size is often, let's say, rivaling the size of a brain, or at least a rat's brain. Moreover, temporal resolution, of course, is restricted in the usual set of trade-offs. There are more fundamental sort of issues, I would say, associated with MRS. So when we do those experiments, we're actually mapping the distribution of a metabolite or of a particular spin. Subtle differences, like where exactly that metabolite is it on the cytoplasmic side or the extracellular side of a membrane? These are essentially impossible to detect. At least people haven't come up with ways to do that. So to detect the kinds of dynamics that we most want to access when we study neural systems, I think those techniques are not going to be that great. There are slow changes In the balance between metabolites that are associated with neuronal activity, those can be detected, but they're generally operating over kind of a minute's time scale, and generally low resolution. So, yeah.
Just regard to the kinetics of the binding and unbinding of the sensors, does that raise issues regarding, well, the process of reuptake of the neurotransmitter, if it's bound? And, you could imagine, for example, that that they could interfere with one another, or if they don't, and the sensor is slow, then you wind up detecting a lot of sensor that, neurotransmitter that's now within the cleft.
Sure. Yeah, so we made stop flow measurements also of the serotonin binding and unbinding. And the assumption that we are making now is that that process reaches fast equilibrium compared with the uptake process. And that's experimentally, supportable in the sense that if we look at the signal time courses, the changes that we see that we wish to attribute to serotonin reuptake are quite slow. They're minutes long. Compared with the binding and unbinding, which is less than a minute. I mean, it's sort of tens of seconds at worst. So we're making the same assumption that Michaelis and Benton made when they first developed their enzymatic model, fast pre-equilibrium, before turnover. In this case, turnover is reuptake, and I think that that's supportable in this case. As you can imagine, well, we could actually put in more parameters in these models, complexity, less reliability, and so we're not doing that now, but we could take a look at it. We did do one important experiment, by the way, which is that we dumped fluoxetine, Prozac, in to inhibit serotonin reuptake, and we saw, in fact, a predictable influence on on the apparent reuptake of these things. So it does suggest that most of what we're studying is that process, or at least a good chunk of it is. Yeah.
So you went away from the bold signal and back to basically, can we do better than the bold?
Bitter irony. Yeah.
And in that part of your talk, you were talking about the relationship between the neuronal activity and the bold signal, but there's an intervening cell that's an opportunity, I think, and that's, of course, the astrocyte, because no neurons touch the vessels.
That's right, yeah.
And so have you thought about the possibility of using some of the unique astrocytic surface proteins to anchor some of these probes, because that would put them exactly where your signals are?
Yeah, so we don't know yet whether such a thing would be necessary. I mean, the brain, of course, contains 10 times as many glia as neurons. They're everywhere. In principle, if we're filling the whole extracellular space of the brain, or at least the chunk that we inject into or deliver our probe into, we're probably in pretty close juxtaposition to astrocytes as well as to neurons. So, is there some particular value in drawing a straight line from the neuron through a particular astrocyte into vessels? I don't know yet. I think right now, one of the things that we're thinking most about is, well, what is the best biochemical pathway by which to mediate the specific signals that we want to transduce to the neurons, and that might bring us to the kinds of ideas that the kind of idea that you just raised.
Hi, that was really great talk. I'm curious whether there's a spectroscopic change in the dopamine sensor when it binds to the dopamine associated with binding to the human.
There is, and that was actually the basis of the protein engineering. So, one of the sort of limitations of constructing these MRI sensors is that doing especially experiments in vivo or even in cells by MRI is a lot more difficult than doing them optically. And so we actually used, in fact, an indirect optically based readout to do most of our protein engineering. We actually had an optical screen that was used to select for protein variants that bound better. And we measured our initial binding constants using optical titration effects. In fact, these P450s are famous for undergoing optical shifts, exactly. But of course, they're absorbance-based. So the sensitivity for the kinds of experiments that people would like to do in vivo may not be there. I mean, I think it's sort of potentially a frontier, but relatively subtle optical imaging absorbance changes, are not, of course, the easiest thing to detect. And these molecules are not fluorescent. I mean, they're paramagnetic, of course, and the fluorescence that you might otherwise see would be quenched.
And you could certainly imagine trying to detect it with photoacoustic imaging, for instance.
That's true. The amounts, I think, are going to be the issue. And actually, I showed a picture of an ultrasound transducer in the last part of my talk, and of course, that's part of, what one might think about there. Yeah. Yeah.
So aren't you worried that bold signals will form an endogenous source of noise, so to speak? Will you have to subtract this same signal, same stimulus given without the your signal through engineered hyperemia, or do you have a pharmacological way of shutting off the bold signal? How do you avoid interference?
Yeah, well, of course, this is an issue, and actually, I showed one image where I had, or rather, Matul in my lab had infused the CGRP in the presence and absence of L-NAME, which is a nitric oxide synthase inhibitor. Other people have shown that bold signal changes can be virtually suppressed using nitric oxide inhibition. The story is, of course, more complicated than that because there are parallel pathways that mediate vasodilation, many parallel pathways. But to first approximation, and actually really frankly to I suspect, sufficient approximation, inhibiting nitric oxide synthase and inhibiting probably cyclooxygenase activity will be enough to shut off the bold sufficiently to resolve these signal changes. So yes, of course, that was a part of the concept.
Thank you, Alan. That was a very innovative presentation. I was wondering if there was ever going to be a focus on like a multidimensional approach to image the mosaic of the neurotransmitters, as well as retrograde signaling mechanisms like the endocannabinoid system, and then if lipids can actually function as those contrasting agents that you're looking for, like the AA that you mentioned.
Yeah. So with the current state of technology in this field, most neuronal metabolites simply cannot be detected in any reasonable way. Let's say I would not, even if you gave me the $10 million, I would not feel honest convincing a student in my lab to try the in vivo experiments. Case in point, obviously the unengineered version of this protein that we use to make dopamine sensors is a sensor for arachidonic acid. This is an important gliotransmitter. It is part of the, neural image. Exactly, yeah. But the concentrations that are relevant to neuronal signaling are in the nanomolar range. We could not detect them using the sensor. So we need qualitatively different approaches for how to measure such dilute, physiologically present metabolites and transmitters, and so we need different ideas. And this idea of engineered hyperemia was one example, I think, of a qualitatively different idea that obviously makes some sacrifices, but could potentially be used to access a much more versatile panel of signaling processes. And of course, tying into genetic techniques, at least in small animals, will probably be one of the ways that can be played out.
Thank you.
Yeah. Oh, I think there's somebody else there.
Thank you. The image of the cannula in the rat's brain was a little bit unsettling, just slightly. Is there any way to look at either a larger animal model where it's easier to place it properly, such as a circus elephant or a whale? Or have you been able to refine the cannula or get an interventional radiologist, perhaps an interventional radiologist, to place the cannula?
Yeah, well, okay. So, one of the sad things about sticking needles in people and rats is that they bleed a little bit. And in fact, if they don't, there may be a problem. But the unphotogenic aspect of the cannula tract is partly due to a little bit of bleeding around the cannula tract. That's probably mostly due to sticking it in. It may be that with a better neurosurgeon in my lab, we could do better. I actually do have a project on trying to make the cannula of the future, in conjunction, obviously, with using these molecular probes. And, probably the chief limitation really is just covering more of the brain. Because although, obviously I'm let's say, being true to myself, to show you the image of the cannula stuck there, the signal dropout due to the cannula placement is not deadly for that whole volume. Some of it is just signal dropout because of the susceptibility effect due to the cannula. And in some areas, it's completely black. You can't see anything, okay? But in other areas, you can still see it. I don't think that doing the experiments in elephants is a good faith effort to solve that problem. Plus, the amount of money required to get a 9.4 Tesla scanner for elephants would be astronomical. We were talking about connections between astrophysics and neuroscience, and there's one right there. Okay? All right.
Thanks very much.
Thank you.